The first speaker is Professor Patrick Tranel, who is Professor of Molecular Weed Science in the Department of Crop Sciences at the University of Illinois. His research program uses molecular and genomic tools to address weed science issues, and he's internationally recognized for contributions that have advanced our understanding of the evolution and underlying mechanisms of herbicide resistances. Complementing his basic research, Pat collaborates extensively with more applied weed scientists at the University of Illinois and elsewhere, fostering a research program that's timely and relevant to weed management practitioners. Pat?
Thank you, Steve. And hello, everyone. Thank you for tuning into this webinar on this topic of herbicide resistance. In essence, herbicide resistance is really an evolutionary process, and hence the photo of Charles Darwin here on my opening slide. So I want to start by stating the obvious, and that is that, as Steve said, this is a timely topic. Herbicide resistance has garnered a lot of increased attention over the last decade or so, and this has been largely driven by the issue or the problem of glyphosate-resistant weeds. And so, as an example, in the Midwestern U.S., glyphosate or water hemp began evolving resistance to glyphosate. First appeared in Missouri, but subsequently showed up in several other states throughout the Midwest. And at the same time, Palmer amaranth, another weed, was evolving glyphosate resistance in Georgia and several other states throughout the southeastern and the mid-south of the U.S. And as should be apparent from this photo, which is supposed to be of a cotton field, this uncontrolled glyphosate-resistant weeds were major challenges for weed management and for profitable crop production. And in fact, it is these challenges that brought herbicide resistance to the national spotlight, not just among weed scientists that were working in the field, but also in the popular press. And so titles started showing up like these on some of our national media outlets and national newspapers. Now, obviously there's a little bit of hyperbole associated with some of these headlines, but in reading the articles, they were misleading, or at least I felt they were misleading, in the sense that they made herbicide resistance sound like it was a brand new problem that we're just beginning to encounter. And so for those of you that aren't familiar with this topic of herbicide resistance, it's really important to point out and be aware that this era of glyphosate-resistant weeds is not the first chapter in our history of herbicide resistance. In fact, we were dealing with issues of herbicide resistance not long after the introduction of our very first synthetic herbicides. I'm also quite confident that this era of glyphosate resistance is not going to be the last chapter. And in fact, I don't think it's going to be the most serious problem we're facing in terms of herbicide resistance. We're now moving into an era of multiple resistances in our weeds populations, and also increasing occurrences and appreciation, really, of metabolic-based resistance. This last era that we're moving into, Todd Gaines will be talking about in more detail. I'm probably getting a little bit ahead of myself, and so let me back up and start with just a definition of what herbicide resistance is. And there's different definitions out there. All the weed scientists don't agree on the same definition, but this is a definition I like. Stated simply, herbicide resistance is an increase in the frequency of resistance alleles in a weed population after exposure to herbicide selection. And the reason I like this definition is it really highlights the fact that it's an evolutionary process. It's simply the combination of selection pressure and genetic diversity which results in evolution. And in this particular case, the herbicides are providing the selection pressure, the weed populations are providing the genetic diversity, and the outcome is resistance. And to take this a step further, in terms of evolutionary phenomenon, the amount of selection pressure that these herbicides provide is just incredibly immense. When you think about what the farmer's trying to do, they're trying to kill every single weed in their field. Immense selection pressure. We have weed populations that can have very high densities, and obviously these weed populations are found across a great expanse of acreage, millions and millions of acres. So you combine this incredible selection pressure with incredible genetic diversity, and the outcome is some fairly fascinating examples of evolution, fairly fascinating mechanisms of resistance. And that's really the theme that I want to focus on today, is to just highlight some of these fascinating, remarkable examples of evolution that we've observed through this process of herbicide resistance. And so what are the solutions? What are the evolutionary solutions that weeds have come up with to avoid herbicides? In other words, what are the mechanisms of herbicide resistance? And so we can broadly categorize these mechanisms into two groups: target site resistance, which simply refers to the fact that the protein or the enzyme that the herbicide binds changes somehow to eliminate or reduce that herbicide binding or the effects of that binding. Non-target site resistance, Todd will talk about in part 2, and that basically encompasses everything other than the target site type resistance. And so to talk about target site resistance today, I'm going to tell 3 short stories using 3 examples, 3 herbicide target sites, and I've abbreviated them here as ALS, EPSPS, and PPO. And so starting with the first one, ALS, acetolactate synthase. That's an enzyme that's involved in the biosynthesis of a subset of amino acids, so-called branch chain amino acids. There are several dozen herbicides that actually target this enzyme. They belong to 5 different chemical classes that have thus far been commercialized. So the ALS inhibitors, the reason I'm starting with those is because this group of herbicides actually leads all other groups of herbicides in terms of the number of weed species that have evolved resistance. And in fact, the closest, the number 2 herbicide group in terms of resistance is not even close. It has less than half this number of resistant weed species. So this large number of resistant weed species suggests that it's relatively easy for weeds to overcome these herbicides, to evolve resistance to these herbicides. And so why is that the case? Well, it turns out that there's lots of solutions to the problem of ALS inhibitors in terms of target site mutations. And so this is a schematic representation of the ALS enzyme. And there are 8 amino acids that have been found in weeds. Substitution of any one of these 8 amino acids can confer resistance while having minimal effects on the normal catalytic activity of that enzyme. So again, lots of solutions, 8 different amino acid, 8 different mutations that could occur to get resistance. And it's even more pronounced than that. In many of these amino acids, there's multiple substitutions that can occur. And this is most notable for the proline, this proline 197 amino acid. In weeds, we have found, weed scientists have found over 11 different substitutions that can occur, again conferring resistance. So lots of solutions to ALS inhibitor herbicides, lots of ways to get target site resistance. This suggests that this enzyme has a lot of flexibility associated with it. How can there be so many mutations that reduce herbicide binding without affecting the normal activity of that enzyme? And the answer to that question comes from structure analysis of the enzyme bound with the herbicide. And so on your left and right here we have 2 different herbicides, a sulfonylurea and an imidazolinone. And the best way to think about this, how these herbicides bind, is they're not actually binding in the active site, rather they're binding at the door. They're blocking the entrance of the normal substrate into the channel that leads to the active site. And so because of this, because they're binding really at the door rather than in the active site, that means there's more flexibility in terms of mutations around that door, changes in amino acids that will alter herbicide binding, reduce herbicide binding, thereby making a resistant enzyme while not affecting normal substrate binding. And so, again, just to illustrate the vast number of mutations that have been found in weeds, this is a list here. I don't know if you can see it. The font might be too small, but the point is it's just a very long list of various mutations in different weed populations and different weed species that have all been identified and shown to confer resistance to the ALS inhibiting herbicides. So I started off by saying that I was going to talk about the remarkable examples of evolution. And so this evolution here, this example is really not that remarkable. These are all simple point mutations, relatively easy to happen. What's really remarkable about this is how many times and how many times it's played out over multiple weed species and multiple populations across a broad geography. So I think it's really an unprecedented example of parallel evolution where all these weeds, these weed populations, basically came up with the same subset of solutions to herbicide resistance. Again, parallel evolution playing out over a blink of an eye in terms of evolutionary time frame, and that this was just a couple of decades when all of these populations evolved this resistance. And so if I can anthropomorphize weeds a little bit, they basically laughed at the ALS inhibitors and said, come on, give us something more challenging. This was easy to evolve resistance to them. And so that brings me to the second story, the second example of target site resistance, and that is to to glyphosate, which is the active ingredient in Roundup, which targets the enzyme EPSPS. EPSPS is also a key enzyme in amino acid biosynthesis. It's a different subset of amino acids, the aromatic amino acids. Despite the fact that glyphosate's been used even longer than the ALS inhibitors, there's only 35 weed species, much fewer weed species, about a tenth, or much fewer weed species are resistant to glyphosate relative to the ALS inhibitors. And you can see that here in this graph of the chronology of glyphosate-resistant weeds over time. And there's obviously two phases in this curve. Initially, for about the first 20 years after commercialization of glyphosate, no glyphosate-resistant weeds were observed, which this lack of observation of glyphosate resistance over this 20-year period suggested that it was harder for weeds to evolve resistance to glyphosate. Of course, the shift in this curve coincides very nicely with the commercialization of glyphosate-resistant crops. And so basically what that allowed, or what that caused, was a great ratcheting up of the level of selection for glyphosate resistance. These glyphosate-resistant crops allowed farmers to use just this herbicide, and to use this herbicide year after year if they wanted to, again, just ratcheting up that selection pressure. So in terms of looking at interesting examples of evolution, this kind of sets up an ideal case study. We have a herbicide where it's hard to get resistance based on the first 20 years of observation, but then we've overcome that difficulty by ratcheting up that selection pressure. So what sort of solutions should the weeds come up with in terms of to escape glyphosate? Before answering that question, let me say just a little bit about why it's harder to get resistance to glyphosate in terms of target site mutations. So this just shows the reaction catalyzed by EPSPS, the enzyme. It produces EPSP using shikimate 3-phosphate and phosphoenolpyruvate as substrates. And again, based on information coming from structure analysis of the herbicide bound with the enzyme, this schematic here is showing the glyphosate molecule here in the bold, and this schematic just illustrates all the different bond interactions, hydrogen ionic bond interactions that are taking place between the amino acids and the herbicide, glyphosate. And so unlike the case of ALS inhibitors where the herbicide is blocking the door of the enzyme, here the glyphosate herbicide is intimately associated with the active site. And you can see here with comparison the substrate phosphoenolpyruvate, and so glyphosate mimics this substrate. It actually mimics this a transition state analog of the substrate, this unusual oxocarbenium ion. And so you can see the very close similarity between glyphosate molecule and the substrate. And so it's not hard to imagine that any change in the amino acids that's going to alter or reduce binding of the binding affinity for glyphosate are probably also going to reduce binding affinity for the substrate, leading to a less effective or maybe even a non-functional enzyme. And so that's the challenge and the difficulty in trying to get target site resistance to glyphosate. And so we knew this, and more specifically Monsanto scientists knew this. They were working away several years ago trying to come up with resistance to resistance to glyphosate through EPSPS mutations. And they had a hard time doing this. They were not able to find any really particularly effective mutations. The best mutations they could find were all in the proline, this specific proline 106 residue, but none of these mutations were great. They caused either modest increases in the Ki for glyphosate, in other words, modest decreases in glyphosate affinity, and or were accompanied with increases in the Km for PEP or a decrease in affinity for PEP. So they didn't substantially increase with resistance without also hurting the normal property of the enzyme. And so in fact these mutations they found were the best ones they could find, the best single point mutations in the enzyme, but none of these were good enough to lead to a commercial crop. So Monsanto did not move any of these mutations forward because they did provide a high enough level of resistance. Nevertheless, what's interesting is when the first weed evolved resistance, target site resistance to glyphosate, it was found to have the same mutation. So it also mutated the proline 106. So nature repeated basically what Monsanto scientists were doing in the lab. Nature repeated out in the field. And it wasn't a great mutation like the Monsanto scientists found. It didn't confer a very high level of resistance, but it was the best that Monsanto could come up with. It was the best that nature could come up with initially as well. And so it wasn't just goosegrass. Several other weeds now have evolved resistance to glyphosate through EPSPS mutations. In all cases, when there's a single mutation conferring resistance, in all cases we've seen in weeds so far, it's always at this proline 106 mutation. So another example of parallel evolution, but at a much smaller scale. But again, it's not a very good resistance mutation, and that's shown here in this dose response curve showing sensitive, resistant, as well as a heterozygous plant, heterozygous for this proline 106 serine mutation in goosegrass. You can see there's an obvious shift to the curve. The resistant plants do have obvious resistance, but the typical use rate of glyphosate is around the 840 gram per hectare rate, and you can see at this rate, particularly if you're in the heterozygous state, it's a very low level of resistance. Better than nothing, so it did evolve, but it's not real high. The photo illustrates that as well. This is a rather low rate of glyphosate, 350 grams. The plants, the heterozygous plant is able to survive, but you can see the great growth reduction and the injury that's there. So, are there other mutations? Are there better ways to get resistance? Well, in fact, there are other mutations in the EPSPS gene that are other mutations that confer very high levels of resistance, but unfortunately, they also confer, they ruin the normal activity of the enzyme because they also simultaneously decrease the binding affinity for the substrate. So Monsanto scientists knew this. They found these mutations. But what they found was by some further tinkering, they could combine a couple of these mutations. So they combined the original proline 106 mutation with this threonine 102 to isoleucine mutation, which conferred a very high level of resistance but also increased Km. But by combining the two mutations, they were able to get high level resistance and no detrimental impact to the normal substrate binding. And so this so-called TIPS mutation, because of the TIPS, TIPS mutation, was actually the first generation of resistance in the Roundup Ready maize. Well, nature came up with the same double mutation solution as well. And so it's just recently reported a goosegrass population that also has this TIPS mutation. And so this is really an interesting evolutionary process. You know, if you go back to the threonine isoleucine mutation itself was probably be a non-functional enzyme because of the detrimental impact of PEP binding. So it probably has to evolve secondly. So what probably happened here was the proline 106 mutation evolved first, provided a low level of resistance, but then through further mutation, that second mutation or maybe recombination joined in, and with that second mutation, a much higher level of resistance. So it's hard though. This is a two-step process, unlikely to occur very often. And so if you can't get a resistant enzyme, is there another way around the problem? And in fact there is, and Palmer Amaranth was the first to demonstrate this. And so what Palmer Amaranth did is it amplified its EPSPS gene. And so this photo here shows the DNA of a nucleus of a glyphosate-resistant Palmer Amaranth in blue, and the pink signal that you see there is from a probe for the EPSPS gene. So the plant has multiple copies of the EPSPS gene, apparently randomly distributed in the genome. So here's a schematic of how this works. Here's a nucleus with one gene. It produces a small number of enzymes which can be bound by glyphosate. In this resistant plant, it now has lots of genes, so it produces lots of enzymes. And so even in the presence of glyphosate, there will still be functional enzymes that can do its job. How this happens, and in fact, even what is being amplified and moved around the genome, I'll just summarize by saying it's kind of a black box at this point. We really don't know. We know parts of the amplicon. We know some signatures and maybe some elements suggesting transposon activity, but really we don't know how this is happening. Okay, so that takes me to my third and final story here on target site resistance, which regards PPO. This is a key enzyme for the biosynthesis of chlorophyll and heme. It has a complex mode of action, so I'll just briefly summarize by saying that inhibition of this enzyme by the PPO inhibitors results in a buildup of photoactive porphyrin, which in the presence of light causes membrane peroxidation. Now these herbicides have been used even longer than glyphosate, which have been used even longer than the ALS inhibitors, but despite that, only 9 weed species have evolved resistance to the PPO inhibitors. So there again, that suggests that it must be quite difficult to evolve resistance to these herbicides. And so it shouldn't come as much of a surprise when we saw the first case of resistance to these herbicides. It turned out to be an unusual mutation. And so the other mutations I talked about previously were just simple point mutations. This mutation here is an actual deletion of 3 nucleotides. So this is just a schematic of the gene. It's a long gene, but in exon 9, 3 nucleotides were found to be deleted in the resistant population, and that caused the loss of a glycine amino acid. So we know a little bit about how this glycine amino acid leads to resistance, again through some structural modeling. This glycine at position 210 forms the last turn or caps the turn of an alpha helix. That the alpha helix is not in the active site, but it lies adjacent to the active site. By removing that one glycine residue, as is in the case in the resistant version of the enzyme, it basically shortens that alpha helix by 1 turn, and you can see that here in the overlay. And so this change is subtle enough that it doesn't significantly alter normal binding of the substrate, but it is significant enough that it does reduce binding of the herbicide by about 10 to 50-fold, depending on the specific PPO-inhibiting herbicide. Maybe the more interesting question is, how did this deletion mutation come about? And so closer inspection of the sequence shows that there's a short little repeat here. We call this a microsatellite repeat, and microsatellite repeats are well known because what happens is during DNA replication there is slippage of these repeats, and so there's insertion and deletion of these repeats. And so what you can see is a little repeat. There's a TGG repeat. There's also a GTG. And you can remove either one of these 2 repeats, and the net result is the loss of a GGT codon. So it kind of looks like a little bit of a magic trick there, but the point is it just happens to work. For the weed at least, it's fortuitous arrangement of G's and T's in this part of the sequence that allowed the loss of one of these repeats to lose a glycine amino acid residue. There's another trick that the weed had to do. So the PPO gene, or the PPO enzyme, is typically encoded by 2 separate genes, which are called PPX genes. One of these genes, both of these are nuclear genes, one of the genes encodes for a plastid targeting sequence, which directs the enzyme to the plastids. The other version of the gene encodes a mitochondria targeting sequence, which directs the enzyme to the mitochondria. Now, water hemp, as well as some other species, have a different form of PPX2, which we call a long form, or PPX2L. And so there's basically a plastid targeting sequence appended in front of the normal PPX2. And so translation of that mRNA results in an enzyme ending up in the mitochondria, an enzyme ending up in the plastids, but due to the presence of a second in-frame start codon there, translation from the second start codon results in an enzyme in the mitochondria. And so this is the gene where the mutation is in, and so in this way a single mutation in one gene was able to confer resistance in an enzyme in 2 separate compartments. And so these sort of evolutionary gymnastics that water hemp had to go through to become resistant gives us at least one explanation as to why resistance to PPO inhibitors has been so rare. And so with that summarizes what I wanted to say about target site resistance. We can see that there's different types of mutations, point mutations, double mutations, deletion mutations, as well as gene amplification. Lots of different solutions that weeds have come up with to try to overcome these herbicides. This is just the tip of the iceberg though, and Todd is going to talk next about other types of changes that can also confer resistance. But I guess before we turn it over to Todd, my understanding is we're going to stop now and answer any questions that you might have.
Yeah, this is Steve Duke. Just a note for the attendees, you can write in questions and we'll try to answer as many as possible. If we don't have time to answer all the questions that are entered into the system here, both Todd and Pat will be happy to try to answer those questions if you'll email them your questions. And one other note too, this entire webinar will be recorded and will go online in about 10 days on the AGRO division website. That's agrodiv.org, the agricultural ag chemistry division of the American Chemical Society. Since I don't see any questions yet, well, here we have. We do have a question here from Bill Venzel. Does PPO resistance confer any fitness penalty, Pat?
So the mutation that we just talked about, so there may be other types of PPO resistance, but the mutation that we just talked about, that's been looked at by a couple of different groups, and what's been found is really no significant fitness cost. So I wouldn't say there is no fitness cost. There probably is one, but it's too small to be really significant in terms of taking advantage of as a resistance management strategy.